So 89% is the number, according to the FDA, of oncology drugs that enter clinical trials, but that fail to progress all the way through to FDA approval. So why is this number so high? Most of us believe that a huge reason for this is the failure of the kind of results that we see in preclinical animal models to replicate and translate into human patients. And so what I want to talk to you today about is a platform, a new type of clinical trial that Presage is pioneering with our pharma partners that allows us to derive a huge amount of information out of each individual patient by microinjecting several different drugs and drug combinations into the patient's tumor while the tumor is still in the patient, and also while not exposing the patient to the toxicity that would be associated with systemic delivery of those drugs. But before I delve into the details of how we do this, I want to give you just a little bit of a personal context for why I think this is so important. This is a picture of my grandmother, and if you look on her left leg, I've zoomed it in here, you can see where she has a pretty big scar where a melanoma lesion was removed from her. A couple years after this, her disease progressed, she relapsed, and she was treated. She actually underwent chemo while she was living at our house, and unfortunately just shortly after that, when I was in 5th grade, she passed away. She was treated with a drug called dacarbazine, which was standard of care at that point. And dacarbazine doesn't really do a whole lot to impact overall survival. So you might say, okay, well, look, that's 30 years ago, right? Melanoma treatment, cancer treatment in general has progressed hugely since then. And in particular with melanoma, you'd be right. If you look here, over the past 5 years, there have been 6 drugs that have been approved for melanoma in the US. And really, the most important application of these is looking at drug combinations. But even with all of this and all of the sort of genomics knowledge that we have, if my grandmother were alive today and we were putting her into a clinical trial, no translational scientist at any pharma company could tell you precisely which drug combination she needs to be on using the standard tools that we have available. So let me explain a little bit more about this. The standard of care, and this sort of reflects some of the stuff that Steve started talking about, the standard of care over the past few years in melanoma has really evolved to be a backbone of a combination of 2 drugs, MEK inhibitors and BRAF inhibitors. And this is this really beautiful personalized medicine story that many of you are probably already familiar with, but for those that aren't, I'll give you hum a few bars of it. So BRAF mutations are present in roughly 50% of melanoma patients, and mutations, this one particular mutation, confers a sensitivity to a class of BRAF mutation or BRAF inhibitors. And these drugs work really pretty well. If you look at the complete response line here, and this is just looking at 2 of the sort of leading combinations. One is the vemurafenib and cobimetinib being developed at Roche, and then the other is dabrafenib trametinib, which is now in Novartis. But the problem with these things is that patients develop resistance pretty much uniformly. And within 9 to 10 months, typically on this drug combinations, they'll start to see progression, and ultimately the disease kills them. So what are drug developers doing to address this? Largely what they're doing is looking at drug combinations. In particular, on top of this BRAF-MEK backbone, they're starting to add now combinations with PD-1, PD-L1, and a whole raft of other targeted agents. But again, the problem is that we really don't have a good handle on how to define those patient populations. What do you look for? What genetic signature do you look for to determine whether or not you want to treat that patient? So I should stop here for a second and just come clean with you. I'm a genomics guy. I worked for about 20 years looking at personalized medicine applications of genomics at companies like Applied Biosystems and Celera and Prologen. And to prove my bona fides, I'll tell you, I still cringe when I see left-handed DNA. So let me introduce what we're doing. So what the approach is that we're applying here is very analogous to the old antibiotic sensitivity test. So really simple stuff, not hard to understand. Plate of bacteria, put these little wafers with different antibiotics on there, and it's pretty clear which drugs this patient's bacteria is likely to respond to and which it would probably be malpractice to treat them with. So we're doing something very analogous to that, but with the more complex background of a tumor. And I don't know how well this show up, but hopefully well enough. I'll show you a lot more detail about how we actually assess these, but the concept is still really the same. So what you're looking at here on the left, is there any way of dimming that light on that one? Is that possible? No? Thank you. So what you're looking at here on the left is a dog patient. This is someone's pet who came into the veterinary oncology clinic with a sarcoma. And what you're seeing here is 8 different drugs and combinations that we've microinjected into this dog's tumor. The bit that you're seeing that we actually co-inject a fluorescent dye, and that is the part that's sort of shining through the skin. What's really important about this is it's not just, each of these drugs is not like a spot of drug that we're putting into the tumor, but it's actually a column that we're introducing in a very precise way that transects the whole tumor. So we do that while it's in the patient. We wait 24, 72 hours later, resect the tumor, and then we cross-section and do fairly standard immunohistochemistry kind of analysis. So just to give you a little bit of a flavor of where we are with this, this is the device that we use in the clinic. On the left, you'll see a lymphoma patient that's being microinjected. We have an open lymphoma trial right now. We've been doing this with patients with approved agents only at this point. And then we're also doing some planning work in other percutaneously accessible tumors, things like melanoma, breast, head and neck, and sarcoma. So this is some work that we presented with one of our partners, Takeda, at AACR, and it starts to give you a handle on the fact that this works actually quite well. What we're looking at is an investigational drug that's been dosed in increasing doses, so left to right, higher doses. And what you can see on the top panel, you're looking at 2 different biomarkers based on the mechanism of this drug. So in essence, all we're saying with this top panel is the drug is working the way that we expect it to work. And this happens to be ubiquitin activating enzyme inhibitor, and we're looking at 2 relevant markers there. Probably the more important part is this bottom panel, which is cleaved caspase, many of you might know as a signal for apoptosis, cell death. And so as we increase the dose of drug, you see an increasing zone of response. And much of the data that I'm going to show you today is this sort of qualitative data. All of this is quantitated in much more detail than I have time to go into today, but just to really introduce the idea. Fundamental question about this approach, right, that you all, I hope a lot of you have this question in your head right now, is it at all relevant? When you take a small microdose of drug, and we use about 100 to 1,000-fold lower than a typical systemic dose, and you deliver it directly to a tumor outside the vasculature, and you do it one time, and then 24, 48, 72 hours later, you resect the tumor and do these immunohistochemistry stains, does that correlate at all with what happens when you do systemic treatment? And so this is, I think, a nice example, but we have dozens like this. Let me just explain what's going on here. There are 2 different lymphoma models that we're looking at. The top one is a treatment naive. This one has not seen treatment. And the bottom one, what we did is created a model by exposing it to increasing doses of doxorubicin to basically make a resistant model to doxorubicin. And so what you can see here is on the top 2 panels, you see DNA damage and cell death. You see a pretty strong response, and as you would expect, we have a dox-resistant line. You have resistance here. Important point, right? These are the exact same doses of drug, okay, in a different tumor. And also not that surprising, but still a little bit interesting, is when you look at vincristine, we actually see a profound response with the naive, and then that response really goes away. So that's not surprising knowing what we know about the mechanisms of resistance. What's a little bit more interesting is when we look at mifosfamide, which is a prodrug, or it's a proxy for a prodrug cyclophosphamide that we use. And while we see a response in the top, it's somewhat muted. And then what we see, though, in the resistant line, we actually see an increase in sensitivity. So this is really unusual. Usually it's the other way around. You see resistance to one drug, it often confers resistance to many other agents. So this was pretty interesting. And so we wanted to look and see how does this correlate with what happens when you look at mouse models. So you take a mouse with these same models and grow them up and dose them repeatedly over time. And what we're looking at now is not just like this microscopic section, but we're looking at tumor volume. And so again, dosing over many days, you can see you have varying levels of a response to dox, cyclophosphamide, and vincristine. But when you look at the exposed model, you can see that it's almost complete resistance to vincristine and doxorubicin and a huge sensitivity to cyclophosphamide compared to what you had over here. Most of the work that we're doing now is around drug combinations. I kind of alluded to that earlier. We have 2 partnerships that we've announced. Celgene is an investor in Presage, as well as we've done actually now 2 agreements with Takeda. And both of those are really centered around looking at drug combinations, finding novel drug combinations. So how do we do this? We look, it's a very simple sort of schematic here. We look at drug A at a certain concentration, drug B at a certain concentration, put the 2 drugs together in that same concentration, and then assess, do you have a response that's more than the sum of the two parts? Do you have real synergy, or is it just additivity? And so I'll show you an example of this, and any chance of just taking the light down a little bit more. Thanks. So this is, I'm happy to share these slides with anyone. I think that's available through the program. But looking at, looking for synergistic effects, true synergy is actually very rare. There are not too many instances where we find truly synergistic activity. This is a case where you see a very minimal response with either of the two drugs independently, but then when you combine them, you see this huge increase. And this is a little bit easier to see in graphical form. Just before I explain this, one of the things that we do is we've developed a pretty sophisticated way of analyzing this data. So we go in and we actually classify each individual cell in a scan. And remember, too, that we're looking at multiple different sections in a particular zone of a tumor and multiple zones throughout the tumor to really get a picture of the tumor heterogeneity. So there's a huge amount of data that goes into this. And we're also taking advantage of the spatial information, where the cells are in relation to the site of injection. And so what we get out of this in the most simple form are these radial distance plots. So you're basically looking at, on the left-hand side, right next to to where the needle goes in, and then drug diffuses from there and is ultimately cleared. And so it's sort of a proxy for a dose-response curve. And what you're seeing here is, again, a profound increase in the effects of the two drugs when combined. So let me go back to the melanoma story for a little bit. This is the way that the MAP kinase pathway is typically drawn. And I know you guys probably can't see this there, but it's this very linear thing. You've got, going in and looking at these different nodes of the pathway. And this is nicely drawn because you can then very easily consider, okay, we have a drug, vemurafenib, going in here, and we have a MEK inhibitor that we're going to treat here. And you can start to get comfortable with the idea that, okay, well, if we throw a combination at this, we'll probably be able to address resistance. And this slide is actually a model of how that resistance comes about. We think it's a little bit more complicated than that, right? I heard a quote this morning from one of the fellows at Amgen who said, if we don't understand how the machine works, we can't understand how to fix it. And I think this, our understanding of really how the machine works is actually pretty limited. And so what we have taken is a way of interrogating biology, but by using pharmacologic interrogation, using pharmacology to go in and push and look at different combinations of drugs to see what kind of responses that we can get. This is a really interesting example of the kind of thing that we can do. That's really, you can't think of really doing this any other way. And so what we're able to do in a single tumor, right, is look at 3 different agents that perturb that pathway. So a BRAF inhibitor, a MEK inhibitor, and an ERK inhibitor. And we can look at, in a single tumor, each of those drugs singly, all 3 of the pairwise combinations of those drugs, and the triplet combination, and really understand what the response looks like with those various parts and what are the important aspects to really combat resistance. So you might ask, why don't you just do this in vitro, right? You can do a lot more scale, and there's absolutely a point for that, to be able to look at huge numbers of combinations. This is necessarily, for the time being at least, going to be limited to sort of dozens of drug combinations. And so there's an interesting paper that just came out at the tail end of last year from Jeff Engelman's lab that really describes, I really encourage you to read this paper, it's awesome, but describes this way of combating resistance with combinations and how they screened for this, how they looked at these, and also how they looked at genomics as a predictor of these. And that was, I think, an interesting aspect of this paper, is that there are a lot of things that genomics just didn't predict that you really need to see phenotypically. But one of the challenges with this is that it takes a long time to do this, and the authors address this in their paper. It takes about 2 to 6 months to be able to really get an answer for this, and that's just not amenable for a clinical trial timeline. The other thing that's a big deal is there's a lot you lose when you're not in an authentic human tumor, right? In a petri dish, there's no immune system, right? And with all of the kind of drugs that we're looking at now, with a lot of the immune oncology agents, that becomes essential. Excuse me. So here's, again, going back to our dog friends. This is a canine sarcoma, and we've looked at a number of different drugs and combinations in here. And I see that the histology slides don't show up too well, so I'll zip through it. But basically, we're able to look at typical pharmacodynamic markers, but then also start to look at immune infiltrates. So we can stain for macrophages and neutrophils and see how do these drugs perturb the way cells are dying in a way that provokes the sort of immune response that you're hoping to see. We can also compare drugs in the same tumor, again, doing the same sort of approach. And looking here at doxorubicin versus mifosfamide, this is not surprising that we see a much more profound T-cell response with doxorubicin in the way that it kills cells. So this is like, this set of data right here that I'm going to show you is probably the coolest data I've seen in like 5 years. So if you're sleeping, wake up to this one. This is cool. So in this case, we delivered an investigational drug, and we had the real fortune of being able to have an antibody that was raised to the drug and the target adduct. So we can see exactly where the drug grows. We can actually even quantitate it. So, no question, we got the drug there. And then we look at cleaved caspase, which is, again, the marker for cell death, for response. And you say, OK, yeah, there's a response there. And you can sort of tick the box and move on if you wanted to. But when you look at this, you can see it's kind of a mottled appearance. There's some cells that are just not responding there, that are pretty clearly resistant. So what's going on there? Maybe you're just not inhibiting target. Well, we've already shown you've got tons of drug there. And in this slide, you can see we're really knocking down the target pretty heavily. So it's not a question of target engagement. We're really doing that. The team at Presage came up with the idea of looking at hypoxia as a way of looking at lack of oxygen and seeing whether that had something to do with it. And what's interesting, when we stain for this protein called GLUT1, we can see that the cells that light up that are hypoxic are almost exactly complementary to the cells that were resistant to, or that we were seeing a response to. And think about this just for a second, right? The typical approach would be, okay, if you're delivering a drug through the vasculature and you've got regions that are hypoxic, that are low oxygen, those are going to be the same regions where you don't have a drug getting there. That's not the case here. We've saturated this, we've marinated this with drug, and you're still seeing resistance. So this is the kind of thing that our partners are able to see and understand about the way their drugs are responding. We have a mantra at Presage, and it is that the only model of human cancer that really matters is the human patient. And imagine being able to conduct a clinical trial where you can look at several different drug combinations, and from that, be able to derive information about sensitivity, resistance, understand how the microenvironment impacts drug response, be able to understand how the immune system is responding to the treatments that you're giving. So this is the kind of thing that we're doing, and we believe that this work ultimately will result in faster, more durable FDA approvals for drug combinations in cancer. And we can't wait for that day.